Well, good day. Hi, I am George Wimberly, Director of Professional Development at the American Educational Research Association, AERA. I'm pleased to welcome you to today's workshop, Modern Meta-Analysis Research. This workshop is part of the AERA and Inter-University Consortium for Political and Social Research, ICPSR, and the Partnership for Expanding Education Research in STEM, what we refer to as PEERS Data Hub. This is funded by the National Science Foundation. The PEERS Hub is a web-based platform, a convening place that enables discovery through wide-ranging forms of education and STEM-related data. It connects researchers to networks for collaboration and communication, and promotes capacity building in research methods, data use, and responsible conduct in diverse forms of data. I encourage you to visit the PEERS Hub platform and become a part of the Hub community. The PEERS Hub website is simple. It is peersdata.net, and we will put that into the chat. Our workshop today is part of a series in collaboration with the Institutes in Research Methods, which, like the PEERS Hub, is part of NSF's Building Capacity for STEM Education Research, or better known as the BURSER program, BCSER. In this method series, the AERA ICPSR Hub features the 11 institutes that seek to promote research expertise in applications of computational, quantitative, qualitative, and evaluative research methods useful to STEM education researchers and broaden the STEM professional workforce. This is our first workshop in this series, and additional workshops will follow throughout the year. Our next workshop actually is in a couple of weeks on February 25th. This workshop is Cutting Edge Quantitative and Computational Methods for STEM Education Research. It will be led by Guanghao Li of University of Chicago and Kenneth Frank from Michigan State University. You'll receive an email message soon to register for this workshop. Now, I know today we've had about 3,000 people pre-register for this workshop from around the world, and it looks like there are right now well over 1,000 people who have already registered. I want to welcome you and really thank you for being here today. You will notice that we are have an American Sign Language interpreter who is providing services for this course, and that there's also a link for closed captioning. So if you check that, if you're looking for ASL or closed captioning, those are both available, and we thank our interpreters for providing those services. Earlier today, you should have received from me an email message that gives you a link to the PowerPoint slides that the instructors will use today, so that those are available to everyone for easy download. Because we have such a large group of people participating in this webinar, I'm going to ask that we refrain from using, I'd say, the chat box for idle chit chat. But as our questions come up, I ask that you use the question and answer box. I will be at designated times going through that and asking some of your questions to our instructors. But following the course today at 3:30 p.m. Eastern, our instructors will have an informal discussion and a question and answer period then. And what we'll do is we'll put a link into the chat box at that point and on the screen where you can go into a meeting mode, and you'll be able to ask your questions directly and have conversation with the instructors. Also, as part of this registration, you will have access to the course recording, and this will be available in a few weeks on both the AERA website and in the PEERS Data Hub. Okay, well, so today's workshop is led by a stellar faculty, and it includes Dr. Terri Pigott from Georgia State University, Dr. Joshua Polanin, and Dr. Ryan Williams, who are both from the American Institutes for Research. We are grateful to the faculty leaders for sharing their expertise with us throughout this course series. We are pleased to offer this workshop, Modern Meta-Analysis Research. Versions of this course at both the introductory and advanced levels have been integral to the AERA professional and development training program and curriculum for several years. The faculty are all experts in systematic review and meta-analysis. They've given similar courses during the AERA annual meeting and in person. Also, last summer, they participated in they led a virtual course that focused on meta-analysis. And for the 2021 virtual annual meeting in April, they will also lead a course on meta-analysis. These are all such experts in the field, and also I always want to say they are such good research citizens and that are eager to give their course. I could go on and on about their accomplishments and achievements, but you can also find their CVs online, and you will see not only their achievements, but also the numerous publications they have in academic journals. Well, with that, I am going to turn things over to Dr. Terri Pigott.
Hi, everyone. Let me get my presentation going here, and we'll get started. Okay, so let's see. Okay, hope everyone can see that. So again, I'm Terri Pigott. I'm at Georgia State University in the School of Public Health and the College of Education and Human Development. I'm joined by my colleagues Josh Polanin and Ryan Williams, who are both principal researchers at the American Institutes for Research. Just a quick acknowledgement. We have funding from the National Science Foundation for our Modern Meta-Analysis Research Institute, which is a training grant on meta-analysis. And here is the grant number. Also like to thank AIR for their contributions, and obviously AERA, ICPSR, Peers Data Hub for hosting this webinar, and our training coordinator, Jay Morris, for his assistance. So the overview for today is we'll be discussing systematic review and meta-analysis in 3 parts. I'm going to do the first part, where I'm going to do a discussion of the systematic review process. Josh will then take over and talk about effect sizes, and then Ryan will talk about synthesizing effect sizes using meta-analysis. So I will go ahead and get started. So my goals for this part are just to sort of give you an understanding of what the goals of a systematic review and meta-analysis are, then to learn the initial steps of a systematic review and meta-analysis, from problem formulation through coding eligible studies for the systematic review. And then Josh and Ryan will take over to talk about effect sizes and basic meta-analysis. So what are the goals of a systematic review and meta-analysis? So I want to start by talking a little bit about the terms that we use, these terms are it's a good chance for us to sort of get an idea of what we are talking about and get on the same page. You hear typically all these different terms referred to in the literature. We hear research review, we hear literature review, we hear research synthesis, we hear systematic review, and we hear meta-analysis. So just want to give you a sense of how I think about this, how we think about these terms. I usually reserve the term literature review or research review to talk about sort of general sort of a more general term to refer to efforts to just review studies on a topic. And I kind of use that term when we think about the typical literature review that might occur at the beginning of an empirical study. And I reserve the terms systematic review and research synthesis. I think about those as synonymous. And I reserve those terms to think more carefully about a systematic and replicable search for a set of studies on a, for a set of studies on a given topic. And it's a systematic and replicable search for a set of studies and a systematic and replicable analysis of those set of studies. And then I use the term meta-analysis to refer to methods for combining quantitative results from a set of studies. Okay, so what is a systematic review or research synthesis? So this definition is coming from an early book on systematic review and systematic review in the social sciences by Mark Pettigrew and Helen Roberts. And you can see here on the slide that a systematic review is a review that strives to comprehensively identify, appraise, and synthesize all relevant studies on a given topic. And systematic reviews then are a method for making sense of large bodies of information and a means of contributing answers to questions about what works and what doesn't, and many other kinds of questions that we'll talk about in a minute. Okay. So again, the goals of a systematic review are to, in essence, summarize existing empirical research, to inform policy, to improve practice, and to provide directions for new research. And at the same time, what we're trying to do in a systematic review is also to limit the bias in the identification, in evaluation, and synthesis of those relevant studies we find to address a given research question. And then meta-analysis, again, is what I refer to as a set of statistical techniques to synthesize quantitative results from a set of studies. And these methods include methods for estimating an average effect and the variance of that average effect from a set of studies. Also methods for exploring the variation across study results, and methods to examine the potential risk of bias in a systematic review. And note that we might use meta-analysis outside of a systematic review. And also note that there are other methodological techniques for synthesizing other kinds of data from a set of studies, like qualitative data. And qualitative synthesis is an active area of its own. And in fact, there is a funded NSF training grant that looks at methods for qualitative synthesis. But that's not what we're going to talk about here. So. Okay, I wanted to also just give you a sense of other, there are other related methods in systematic review. You might hear the term scoping reviews. And scoping reviews are aimed at identifying the size of an evidence base. How many studies are available in a given area, for example. So checking in and checking to see how much literature is out there, how many studies have been conducted in a given area. So that's what we usually refer to as a scoping review. And related to that are also things called evidence and gap maps. And these evidence and gap maps provide a more formal description of a set of studies or the evidence base available on a given topic. So in this next slide, I'm just showing you a quick, it probably will be hard to see this, but I'm showing you an example of an evidence and gap map published by the Campbell collaboration. And this is looking at interventions that help, that are in the general area of disability research. And you can see that the rows of this evidence gap map are different interventions. And the rows that I have shown you here are interventions focused on health promotion, prevention, and rehabilitation. And the columns are outcomes, also related to health. And you can see the first, so if you look at the upper left cell, we're looking at interventions that are aimed at promotion of health and that are measuring mental health and cognitive development. And within that cell, we see a bunch of bubbles, and those bubbles indicate the size of the evidence base. The yellow and blue bubbles are indicate numbers of impact evaluations conducted on health promotion and focused on mental health and cognitive development. And the red and green bubbles are looking at reviews. And you can also see the size of the bubble indicates the size of the evidence base, and also the colors indicate quality of those particular either reviews or evaluations. So this is another sort of related area to systematic review, and you can also see other examples on the Campbell website, and the link to this particular evidence gap map is given in the upper corner. Okay, but back to our matter at hand, systematic reviews. So let's talk a little bit about the steps of a systematic review. So these steps come right from work by Harris Cooper, and you can see that we start out, as we do in any empirical study, with problem formulation, with formulating a question for a systematic review and meta-analysis. Our second stage is then searching the literature, so finding studies that are relevant to our research question. Then we need to screen the literature. We need to look through our literature search to find the studies that are in fact relevant for our particular question. The next stage is coding study characteristics. So identifying and looking at extracting information from each of the studies, and we'll talk a little bit about that in a second. And I will be covering those first 4 steps. Also part of coding study characteristics is computing effect sizes, which Josh will talk about, and then analyzing and integrating those outcomes of studies, which Ryan will talk about. And we won't talk in this very short webinar about interpreting the evidence or presenting findings. So we're just let's go ahead and start with our step 1, problem formulation. So given that we are all funded by NSF here, we're going to focus, my examples are going to focus on STEM education research. So there are sort of 2 families or general questions that we ask in a systematic review and meta-analysis in STEM education research. One is about intervention effectiveness. So how effective is an intervention or family of interventions in a given context? And so I'll give you a couple examples of that. So this is a very common question that we ask in a systematic review, asking about whether or not a given intervention is effective and under what conditions that intervention is effective. Another set of questions that are also important in STEM education are questions about associations. So questions about how 2 constructs might relate to one another. You know, what's the correlation between two given characteristics or so forth. So we're going to look at examples of both of those 2 questions. And these cover most of the kinds of questions that we might ask in a systematic review and meta-analysis in STEM. But other questions can also be addressed in a systematic review and meta-analysis. And these questions tend to be more common in health and medical research. One is about prevalence. So looking at the literature to try to estimate the occurrence of some condition. So that's another question people might address in a systematic review. Questions about diagnostic or prognostic tests. How well does a given diagnostic instrument or test identify a condition or predict a condition? Is another set of methods we might use in a systematic review and meta-analysis. And also a common question asked in many health and medical systematic reviews is about comparative effectiveness. And these are systematic reviews that are really directed at head-to-head comparison of 3 or more interventions. So here's an example to go back to our context of STEM education. Here's an example of a systematic review and meta-analysis focused on intervention effectiveness. So this one was published in 2017 by Brian Belland, Andrew Walker, Namju Kim, and Mason Leffler. And it looks at, it's synthesizing results from research on computer-based scaffolding in STEM education. So their particular research question is, what is the impact of providing computer-based scaffolding to students engaged in ill-structured problem solving in STEM? So their question is really about the effectiveness of computer-based scaffolding. So this is a classic intervention example for systematic review and meta-analysis. A related type question is on the following slide, also a paper published in the Review of Educational Research, looking at generational differences in academic achievement among immigrant youths. And so in this particular systematic review, they're not comparing two interventions. So we're not comparing a treatment and control group. We're not looking at the effectiveness of an intervention, but instead we're comparing two groups. And in this case, the achievement of first versus second generation immigrant youth. So this is another sort of version of a kind of question we might ask in a systematic review and meta-analysis. And then our other major question, again, is about associations. So here's a third systematic review published in the Review of Education Research. And this one now is looking at the relationship between mathematics anxiety and mathematics performance among school-age students by Jessica Namkung, Peng Peng, and Jin Lin. And here the question is, really, what is that relationship? How strong is that relationship between math anxiety and math performance in school-age children? And also they were interested in identifying potential moderators and underlying mechanisms of that relationship. So here's an example of a systematic review where the question's really about associations, not about interventions or about differences between two groups. Okay. So in addition to one way that we talk about developing a question for a systematic review and meta-analysis is to use these particular frameworks to help us outline the aspects of that systematic review and meta-analysis. And there are two major frameworks we use that are really directed more at effectiveness or intervention systematic reviews. And these are the PICOS and the UTOS framework. And these frameworks help us to outline the critical aspects of the research question that will guide the systematic review and meta-analysis. And they'll also be helpful to us when we are beginning to try to identify the studies that are most relevant to our research question. So the PICOS framework, I'll start with the PICOS framework. Again, these are sort of more directly focused on questions, intervention questions about effectiveness. But the PICOS framework consists of P for population or participants. So who are we most interested in studying in our systematic review? I for interventions, if we are doing an intervention or effectiveness systematic review. C for comparison group. So what kind of comparison group is most relevant for our particular systematic review? And you can see where this is more directed at interventions. Outcomes. What are the primary and secondary outcomes that are of most interest in our systematic review? And then S is typically study designs. What study designs are going to be most appropriate for the question we're asking. And sometimes we might think about, some people think about S as also settings, if that's a relevant piece of your systematic review. We also have another framework, which is based on Cronbach's UTOS. Some people find this helpful too. U for units or subjects or participants, who are the people that are most important to think about in our systematic review. T for treatments or interventions. O for outcomes, and S again for settings or study designs. And you can see that the UTOS is also more focused on interventions. So if we just go back to our example, our Belland example on computer scaffolding, we can see that the units or subjects for that particular case was any students from K through college. The treatment or intervention was a computer scaffolding intervention. The outcome, they had a variety of outcomes that related to STEM education. And the study designs they were most interested in were randomized controlled trials. We might also adapt PICOS for those association systematic reviews. In that case, we might think about P as, again, populations or participants, and I maybe we might change that to be independent or predictor variables. So for the math anxiety and math achievement systematic review, our I might be, in essence, math anxiety and math achievement. Conditions or settings, in this case, that was that particular systematic review focused on school-based contexts. The outcomes, they in that review, they lay out the different ways that they measure math anxiety, and study designs for that review were also pretty broad. In general, we use PICOS just as a heuristic to help us, lay out for our systematic review and meta-analysis question those really key aspects of that are going to be important as we begin to search for studies. Okay, and then a couple of just additional considerations when you're thinking about developing your research question for your systematic review. One question is sort of how mature is the database? Are there sufficient studies available to you to conduct a systematic review? And what I mean by that is if, you're interested in studying a brand new intervention, that was just developed, this might not be the time to do a systematic review. And so as you're thinking about, developing a research question, you need to think about sort of how much literature really is out there. Is it the time to do this kind of review? It's also important to think about how a systematic review might fit into the current evidence base. So, what are the controversies in the field? What are things that are still needing to be illuminated? How would a systematic review then help us move that field further ahead? What are the questions that we might want to really think about as we're developing that systematic review question? And then a third issue that always comes up in systematic review and meta-analysis is sort of an understanding and a deep understanding of the content area and the concepts of interest in that systematic review. So as you're beginning to think about, developing your systematic review question, you need to have some deep content knowledge and understand how the field has been thinking about these concepts of interest. So for example, in the Belland computer scaffolding literature, how do we think about computer scaffolding? What does that mean? In the other review on mathematics anxiety and math achievement, how are we defining, how is the field defining math anxiety? So these are sort of deep content questions that you need to kind of grapple with as you begin to think about your research question for a systematic review. Okay, so once you have developed your systematic review question, the next big stage or step 2 of the process is to find all relevant studies in the area. Okay, so our inspirational goal is always get all the studies that have ever been done in a particular area that's relevant to our question. And our realistic goal is to actually get as close to a representative sample as possible and to make the case that we have done that. And so in order to make the case that we have done a good job of searching the literature, we need to search the literature using a number of strategies. This means searching multiple electronic databases. This means also using other kinds of strategies outside of using electronic databases for studies. For example, forward citation searching, what we call hand searching, which is looking at just the list of articles published in particular journals, and other kinds of strategies. And it also means searching for unpublished literature. And I'll talk about that in a second. It's also important to remember that the search strategy is part of the key methods for systematic review, and those methods then should be transparent and replicable. Okay, so in the literature search, we have a number of potential sources of bias that might impact the results we would have in a systematic review. So for example, we might be missing relevant studies because we did a limited search strategy and we failed to consider multiple and related disciplines. All of us are living in a multidisciplinary world. Most of the questions we address in STEM education are also addressed by others in other fields. And so it's important to recognize that we need to search multiple online databases that reach different disciplines. And we also know that the way information is cataloged and indexed and so forth in the world, it also means that we need to search multiple databases because they don't all overlap. And it also turns out that many of us have access to different forms of those databases. A second potential source of bias that might creep into your literature review stage is publication bias from searching and including only published studies. We don't have time to go into this here, but it is well established that in the social sciences, in psychology, in medicine, studies that are statistically significant are more likely to be published. And if we only focus on published studies, we are likely to overestimate the impact, for example, of an intervention. So we need to search unpublished sources as well, and I'll talk about that in a second. Another way bias creeps into literature review part is our literature search stage is that we might use search strategies that fail to represent sort of how concepts are defined or operationalized. And again, that goes back to that point I made a few minutes ago. It's important to understand whatever concepts we're using in our systematic review, that we understand how they are defined, how they are operationalized in different disciplines, so that we can capture that how all those concepts are thought about across the different disciplines. And again, you know, another potential source of bias is if we have a non-transparent or non-reproducible search strategy. Okay, so the most important advice I can give, and that we give to anyone who's doing a systematic review in or meta-analysis, is get help from a search specialist or a librarian. We are not, there's a lot to know about how to search. And so our best advice to you is to find someone who can help navigate the multiple databases and search strategies for systematic review. If you're lucky enough or privileged enough to be in a big university system or institution, you may have a lot of resources around that. So for example, my own institution at Georgia State has a whole page and bunches of resources on systematic review, and I think it's open to this page, for example, is open to anyone. And I'm very privileged to be able to have lots of librarians who can help me out here. So again, going back to those sort of three general strategies for a literature search, you know, you want to, first of all, search in online databases and multiple online databases, psychology, education, and ERIC. There are many contexts in which we might find studies also in PubMed, and all of these online databases contain some gray literature. We also want to search directly for gray literature using methods like searching for dissertations in ProQuest, using ProQuest, or also searching for studies conducted by research firms. So my colleagues, both Josh and Ryan at AIR conducts a lot of primary studies, and so those are also sources for unpublished data. And then use other search strategies like forward citation searching, checking the reference lists of systematic, or contacting researchers who are active in the field. Okay. Just a quick note on documenting and organizing the lit search. I think by now you understand that this is a very complex part of a systematic review and meta-analysis. So if you're not using a reference manager, use a reference manager. So Zotero is one that's free. Some of us have access to EndNote or Mendeley. All of these search reference managers allow a download of your search results and then help you organize all the multiple searches you're going to be doing in a systematic review. You'll need to make sure you document aspects of that search because not only to keep track of it for yourself and also to report it out as part of your systematic review when you're publishing your systematic review and meta-analysis, but also to have to document it in a Prisma flowchart, as I'm going to show you in a minute. This is the Prisma flowchart. I've given you the link up here. The Prisma flowchart documents in a transparent way what your literature search was like. And you can see here, it asks you to document how many records you identified through database searching, how many through other sources, and so forth. I have an example of a completed one here. This one was done for a Campbell systematic review, which is openly available. This one is on targeted school interventions for improving reading and math for students who are facing academic difficulties. You can see here that they identified 17,000 records and another 7 using other strategies. They eliminated 24,000 that were duplicates, and eventually they got down to 71 studies included in their meta-analysis. Okay. I have 2 references here for you that might help your literature searching. The Campbell collaboration has an informational retrieval guide that's freely available. The link is here. It's under the methods guide in the middle of that page. And although it's focused on medicine, the newest Cochrane handbook, chapter 4 in the appendix, has a lot of resources for thinking about developing your search strategy for an online database. Okay, so moving on. So now we're going to assume that you've completed your literature search and you've got all your studies ready to screen. So the goal of the screening stage is to identify those studies that are relevant for your question. And these studies are those that meet those PICOS and UTOS criteria that we talked about just a few minutes ago. We usually screen for systematic review in 2 stages. The first stage, we screen just the abstract and title, and the second stage, we use the full text screen. We screen the full text of what we think are potentially relevant articles. So title and abstract screening, you might think, wow, given that many times the abstract of any primary study bears only passing resemblance to what's in the study. In fact, it turns out that screening titles and abstracts can be is really an effective way of screening out, completely irrelevant literature for your systematic review. The link at the middle there is a link to an open access article written by Josh that talks about best practice screening. But basically, best practice is to use a very short screening tool with 3 to 5 criteria based on your PICOS or your UTOS. And then you just make decisions based on the titles and the abstracts. And in the stage, we also are generous. If we can't figure out if it's relevant or not, we keep them. There are also other kinds of, to conduct that kind of screening, people have used their reference manager, but there are a couple of tools out there like Abstractor and Rayon, which are freely available screening tools directly for systematic reviews and meta-analyses that can help with screening. They use a machine learning algorithm that helps you to organize and also keep the screening going a little bit faster. So we highly recommend the use of those tools. Then once you've screened titles and abstracts, the next thing is to actually go in and pull the full text of those studies you think are going to be relevant for your review. And while abstract and title screening can help narrow some of the studies, and it's really good at finding out what studies are actually empirical studies and what studies might be more essay type studies. There are some eligibility criteria for your systematic review that are going to be difficult to find in an abstract or title. For example, study designs. If if you're really interested in only using randomized controlled trials in your systematic review and meta-analysis, that kind of information is harder to find in an abstract than other types of information. So again, we get the full text PDFs of studies, and we do another round of screening using similar steps that we use in abstract and title screening. So a small 3 to 5, item screening tool based on PICOS and UTOS to try to get to our final or near final set of studies relevant for the systematic review and meta-analysis. Okay, so there are a couple potential issues or biases that might occur when we're in this stage. And obviously the biggest one is that we've incorrectly labeled a study as eligible or not eligible. So a couple of strategies to to avoid that bias. One is to apply a transparent set of criteria and apply those consistently across those screening stages. So in the middle of screening, we don't want to be changing our eligibility criteria, for example. And then as we're screening, we want to make sure that we are interpreting those items consistently across screening stages. As you can see, the example I just showed you identified 17,000 plus studies. So you can see if you're screening 17,000 studies, you're going to get a little tired. So one of the things that that is also best practice is that you independently and double screen all identified studies. Okay, so as we're going through our whirlwind tour, we are already on step 4 of the systematic review and meta-analysis. So once we have our research question, we've done our literature search, we've screened our studies, and now we have this set of studies that we think are most relevant for the question that we're interested in. And so in this stage, we are interested in what we call study coding or data extraction. So there are a couple goals that guide us when we're thinking about study coding and data extraction. One of the reasons why we do this is just to gather information to describe the evidence base for the research question. So we want to tell, when we have our systematic review, when we're done with our systematic review, we want to be able to say, there are this many studies that are relevant to this question. They are looking at this set of population that has, these are the participants, these are the settings that we're seeing things in, and so forth. So we're gathering information from studies just so that we can give our audience and the stakeholders interested in our systematic review and meta-analysis an idea of what those studies look like. The second goal of study coding and study, and Josh will talk about that, so I won't spend any time on that now. And then finally, we also want to code characteristics of studies that may help us understand why study results vary. And this is sort of the exciting and one of the exciting parts of meta-analysis, for me at least, is to think about why might study results differ. If we have an intervention and we're seeing that not all the studies are agreeing on how effective an intervention is, what are the potential reasons why? Does it have to do with differences in studies and their participants, in how they implemented the intervention, and so forth? So the other part of study coding and data extraction is to then, think about coding aspects of study design and methods and so forth that may help us explore why study results differ. But I also wanted to put another caveat here, in that this is one of the more systematic review. So although the tendency for all of us is to code everything in kitchen sink, we do want to be strategic in how we think. Back again, question of how the systematic review is going to fit into the literature, and what else we need to think about. And what are the kinds of questions we want to illuminate in our review. Okay, so if we go back to the Belland, the computer scaffolding example, here's just a list of some of the things that Belland and his colleagues coded in that particular review. They looked obviously at education level. So clearly maybe computer scaffolding interventions could differ depending on if you're a kindergartner versus if you're in college. The assessment level, so what were the assessments used in that study at a lower level of concepts, or were they at a higher level of application? And that might, that might be related again to, the effectiveness of computer scaffolding. And I won't go through the rest of those, but you can see that, these things that are coded in that study were both to describe the sets of studies that are available in this topic, but also to talk about why the intervention might be, how effective that intervention might be, and how it might vary across these particular contexts. So where might bias occur in the coding process? So the coding, obviously we might get bias in the coding process through an inconsistent application of our codes or inconsistencies across coders. And so best practice is to, have a clear coding manual, make sure we train our coders, and when possible, all this is again a resource intensive part of a systematic review, use 2 independent coders on each study. And again, it's important to think about the management of this process and using quality checks and regular meetings to resolve inconsistencies across coders. Okay, so I'm about there with my part. So again, what I wanted to make sure we emphasized is that, systematic reviews need to be transparent and reproducible. If they're a tool to synthesize studies and we want to use the results of a systematic review and meta-analysis to improve practice, to improve policy, we need to make sure that we are also transparent and about our methods. Okay, and in that vein, we, and I'm talking about we as a team, Ryan, Josh, and I advocate that all systematic reviews and meta-analysis adhere to open science principles. So we believe systematic reviews and meta-analysis should follow a protocol that was developed in advance. And those protocols could be posted, for example, in the Open Science Framework. Examples of these can be seen in the Campbell collaboration, who have from the beginning always published protocols and completed reviews that are open access on their website. And since the data from a systematic review and meta-analysis is not sensitive, it's from, for the most part, published sources, there's really no reason not to, post both the data and code after the completion of a review. So I want to end by giving you a few resources for the beginning stages of systematic review. I've mentioned Harris Cooper's book. There's also a book by David Goff, Sandy Oliver, and James Thomas that is an introduction to systematic review, and also talks about systematic reviews with qualitative data, and then the classic Lipsey and Wilson. So I am going to stop there and see, George, if there's any questions.
Thank you so much, Terri. That was a great presentation. One of the things that's come as questions seems here is, can you speak a bit more about how many studies are necessary for a systematic review?
So always a tough question.
Yeah.
So facetiously, we always, sometimes we facetiously we say 2 for a meta-analysis, but it's a hard question to answer. But if you look in the literature, and Josh or Ryan step in here too, you'll see that a typical meta-analysis, say in review of educational research, could be 20 to like 50 studies.
In sort of in a similar vein, can you also speak to about how many researchers should be involved in the actual, as you check the reviews, coding reviews?
Oh, when you're coding.
In
Okay,
your coding
so
as well.
I guess I have 2 quick answers for that. One is, many of you on the webinar are probably tired of me saying this, but this is a team sport. Doing a systematic review and meta-analysis requires a team. And it requires a team of people with various expertise, deep content knowledge, people understand meta-analysis, good to have a librarian, and so forth. So in general, you want at least 2 people, helping with the coding and the screening. But as you can see, if you're doing a very big meta-analysis, it would be good to have a bigger team. Josh or Ryan, if you have any thoughts about that too. Okay.
Okay. Don't think they have that. There are other comments there.
Yeah.
Well, in the interest of time, too. One other thing, Terry, if you could just say something about, I know you talk about reviewing abstracts. What do you suggest when abstracts may not be available or are not consistent, within a field or a discipline?
Yeah, I mean, I think that we struggle with that. One thing that, is very clear is that, abstracts are not, not necessarily the best representation of a study. So when you're screening, you are more generous. So if you can't find the information you need in that abstract, you screen it in, and then later on when you're doing the full text, you might be able to find the information you're interested in.
Great, thank you so much, Terri. You're getting the comments in the chat box and the question and answer all just are so positive. I think everyone's really enjoying your presentation.
Thank you.
One other thing that's come up a few times, though, too. Can you also speak about unpublished studies?
Okay.
Where you might find those.
Yeah. So I think it's sort of a high effort, somewhat low yield aspect of a systematic review and meta-analysis, but we do it because this is really the only way to sort of demonstrate and make the argument that we've tried to get unpublished studies in. A good source of those are dissertations. And some people might argue that they're not as rigorously reviewed as published studies, although those of us who've done a dissertation feel that they were rigorously reviewed when we were in the middle of it. But it's also the case that, which I haven't really touched on in this presentation, another aspect of coding that's really important is that we also code for study quality. So published studies, unpublished studies, if people are worried about their quality, another piece of a systematic review and meta-analysis is always to code the quality of a study. And so we kind of catch that, we're able to deal with that issue of study quality in the coding, as opposed to in eliminating those kinds of studies right off the bat. I hope I answered the question there. So.
Thank you. Okay, well, I know that we'll have other times at the end of the session for questions too. So I think right there we can move on to our next session.
Okay.
Everyone, give me 1 second to share my slides. Okay, no rest for the weary, although we are taking a break for a few minutes after my session. So like George and Terri already mentioned, my name is Josh Polanin. I'm a principal researcher at the American Institutes for Research. If you're not familiar, that's a research firm, a US-based research firm, And we do all sorts of social science research projects. And I'm lucky enough to work on basically only evidence synthesis projects. So I spend 100% of my time either thinking about or doing systematic review and meta-analysis. And it's something I really enjoy. So I'm thrilled to get the opportunity to speak to all of you today, and especially grateful to George and the AERA team for setting this up for us. And the last thing I'll note right before I dive in, I've been saying a word of support since this summer, and I've seen no reason to stop saying anything. And I just like to express my support for the Black Lives Matter movement, and I stand with them through and through. So with that, thank you for allowing me a minute to divert attention, but we can sort of dive back in now. So Terri already gave a great summary on the process of conducting a systematic review. And she mentioned right at the end that as part of the coding process, the next along with that stage, we're going to estimate effect sizes. And so that's what I'm going to spend the entire next 45 minutes on. So I'll flip slides here. So let's start here. What is an effect size? What do I mean by that term? And why is it important for meta-analysis? So let's imagine that you're interested in understanding the effects of various programs, all with the same characteristics and maybe core components, and whether or not they have an impact on some outcome domain. And I've got an example here of a STEM meta-analysis that was just published a few years ago, and it's looking at the impact of homework on students' academic achievement in math and science. And the authors say that the research conducted since 1986 were synthesized to examine that relationship and a range of factors that moderated that relationship. So you can imagine in a study like this, in a study that synthesized 30 years worth of studies, that achievement, math and science achievement, is measured possibly quite differently across all of those studies. But underlying all of them, there's that same achievement construct. So we need to find a way to essentially put all of the studies on the same scale so that they can be synthesized in the same model. And to do that, we estimate an effect size. And so that's the expression here that we're using in a meta-analysis, and it's a quantitative expression of the size of the relationship of interest. And so the relationship of interest can vary based on the topic of your meta-analysis. Terri mentioned a number of them at the top of this talk. And Terri, I see that your mic just came back on. Is there something? Did I miss something? Nope. Okay. Just an errant mic. So an effect size is the magnitude of that relationship, and it can vary depending on what you're interested in synthesizing. But the beauty of an effect size is that it is comparable across studies. And so there's different ways of estimating that effect size. We're going to go through 3 of them today. There's actually many, many more than 3. And you can actually estimate an effect size in both an unstandardized and a standardized way. The unstandardized way expresses the magnitude of the relationship in whatever units it was measured in. And it's usually not what we wanted. You're only interested in the unstandardized measure of an effect size if every single study in your meta-analysis measures that construct, that achievement construct, using the exact same test or outcome, which is hardly ever the case. Most often, what we're going to be interested in calculating is the standardized effect size. And that standardized effect size is expressed in some measurement unit, usually the standard deviation. And it's this expression of the standardized effect size that allows us to estimate a meta-analytic model by being able to combine effects from multiple studies. So that's the key here. Every study will produce, that's included in your systematic review and meta-analysis, every study will produce a standardized effect size, and then along with that, the standard error of that effect size. And that standard error that we calculate along with the effect size will be used in our meta-analytic model. And Ryan will talk about how that standard error gets used in those meta-analytic models. So that's the key. If you take nothing else away from this talk today, it's that in order to do a meta-analysis, every single study you have to be able to estimate an effect size and a standard error. And those effect sizes will vary based on what information the authors report. And that variation, so whether or not it's a D, an R, or an odds ratio, that's what we're going to talk about today. How do you estimate those various types of effect sizes in order for those studies to be included in a meta-analysis? Okay. So yeah, what will we cover? The D family, the R family, and the odds ratio family. And really we're going to focus on a couple of primary things. The sort of thing that you're most interested in as a practical meta-analyst is what type of effect size are you going to calculate? And as a result of that type of effect size you're going to calculate, what type of information do you need to collect from the study? The rest of the how the effect size is estimated, how the standard error is estimated, and then eventually how a meta-analytic model is calculated, all those rely on a set of formulas and strategies that have been developed over time. And it's certainly very important to have a good understanding of what those formulas are, and we're going to talk about that in the next few minutes. But from a very, very practical level of you just want to start working on a meta-analysis and have enough information to estimate some effect sizes and then run a meta-analysis. You're really interested in what is the effect size that I'm calculating, and then how do I go out and find the information that I need to estimate that effect in the standard error. And the last thing that we'll talk about today, it's just one slide. And if you're programming averse, you can just sort of shut your eyes and cover your ears for a minute, but I will show some programming script using the R language to illustrate, frankly, how easy it is to estimate the effect sizes with the collected information. Alrighty, take a look at the time here. Okay, so let's start with the D family, and this is the standardized mean difference, okay? So we're interested in the standardized mean difference if we are comparing two groups, and those two groups have an outcome that is continuous, and therefore we can calculate a mean for each group. And so this is the most common effect size that is used when we're looking at experimental groups, like an intervention and a comparison group, or if we're interested in comparing the differences, like I say on the slide here, between boys and girls. Okay, so if you're, to give a concrete example, back to the example the thought experiment I started with, all the studies that were included in that meta-analysis were looking at the effects of homework. So some group of students received some homework, some groups didn't. Those would be the comparison groups. And we'd be estimating the standardized mean difference between those two groups. And that standardized mean difference is generally referred to as the D family in the literature. Okay, So a little bit of notation. I promise there's only a few formulas in these slides. Terry's already mentioned a few times that we have some other training programs that we conduct, both with AERA and for NSF, and we do some deeper dives into the formulas and variations on these effect sizes. So if you're interested in doing a little bit of a deeper dive, we'll post some links to those websites and those training materials. And Terry's already mentioned at the end of her slides some really great resources. The Lipsy and Wilson book in particular has a number of different formulas available just to estimate the standardized mean difference. So there's actually a lot of different ways to estimate the standardized mean difference. But for us right now, we're interested in a pretty simple example. And this is an example where you can imagine let's stick with the intervention and comparison group example. There's one intervention group, one comparison group, and we could represent the intervention group with group one as group one, or G1 here. And the comparison group would be represented as G2, group two. Okay, so keep that in your mind for a minute. And then there's some intervention that's implemented. And at the end of that intervention, we measure some outcome. Maybe it's math achievement, maybe it's some other outcome that you're interested in. And we calculate the means for the two groups, the intervention group and the comparison group. Those are represented by X bar G1 and X bar G2. So those are the two pieces of information that we will be collecting as meta-analysts, the means for each group. Then we also need to collect the group's sample sizes. So how many people participated in the study among each of the two groups. So that's represented by the little n G1 and little n G2. The total sample size is represented by big N, and that's just a sample size for the first group plus a sample size for the second group. And then the last set of information we need are the standard deviations. So we need the group standard deviations. So again, imagine that you've got the two groups in your data set, and you're going to estimate the standard deviation for the intervention group, and you're going to estimate the standard deviation for the comparison group. Okay, so it's those three sets of information: the means by group, the sample size by group, and the standard deviation by group. And that's what you're looking for from every study that's of this type. So what do you do with that information? You put it into the standardized mean difference effect size calculation, and it's fairly simple. It's the mean of the first group minus the mean of the second group divided by what we call the pooled standard deviation. And the pooled standard deviation is just basically an average standard deviation. So we're taking that mean difference, the x bar of the first group, the mean of the first group, and we're subtracting off the mean of the second group, and we're dividing it by that average standard deviation, the pooled standard deviation. And when we take that difference and divide by the pooled sample standard deviation, that puts that mean difference in the scale and the metric of the standard deviation. And when you do that for every single study, every single effect size, now you have measures that are comparable across studies and measures that are allowed to be combined across studies. So that's the power of calculating the effect size. Oh, I'm sorry, skipped too many slides. Excuse me. So that's the formula for the standardized mean difference. There is one correction that we always apply to the standardized mean difference, and it's referred to in the literature as the Hedges G effect size, or the bias corrected estimate. And it's called the Hedges G because Larry Hedges derived it in 1981. And it's a pretty simple formula, actually. All we're doing here is we're taking that standardized mean difference effect size that we just calculated, and we're multiplying it by this little factor here on the screen, the 1 minus 3 over 4 times the total sample size minus 9. And that corrects for small samples. So essentially what's happening is that Larry in 1981 did some calculations and determined that when you have really small samples, so samples less than total 50 total participants, the effect size tends to be too large. So it's biased. And in order to correct that bias, we apply this little correction here on the screen. So if you see in the literature, if you're reading a meta-analysis and the meta-analysts say we estimated Hedges G effect size for each study, this is what they've done. Okay, so you've estimated the effect size. Now there's that one other piece of information that you need to estimate, and that's the standard error of the effect size. And again, the formulas looks a little bit more complicated than it actually is. On the left side there, it's just the total sample size, N1 plus NG2, divided by the product of the two sample sizes, plus Hedges G squared divided by 2 times the total sample size. And that's the effect size's standard error, and it's a measure of its precision, right? And precision is a function of the total sample size. So the larger the sample size, the more precise our estimate of the effect size. The more confident we are that we have estimated the true effect size. And we're going to leverage that piece of information, the precision of the effect size, when estimating the meta-analytic model later on. All right. So I said to you at the beginning, what I'm going to focus on, or part of what I'm going to focus on, is really how do you apply those formulas in practice? So what are you actually looking for? What are you actually doing as a meta-analyst when you know you need to calculate, in this case, a standardized mean difference? So to do that, let's look directly at a study, and I think this is a study of a program called multi-systemic therapy. But you can imagine this to be any type of study that you might be interested in that has 2 groups. In this case, the authors were interested in reducing the number of arrests after the individuals received an intervention, in this case, the multi-systemic therapy intervention. So that orange box that I have outlined here illustrates the information that we would need to collect from the study. So underneath arrests, it says there's a large capital M there, and if we go all the way over to the right, we can see underneath the multi-systemic therapy column, underneath post-test, it says 0.87. So that's the mean number of arrests that occurred after the intervention occurred for the treatment group, for the intervention group. And for the comparison group individuals, the post-test arrests, the average post-test arrest was 1.52. So those are those 2 pieces of information. Then we have the standard deviations to collect. It's 1.34 for the intervention group and 1.55 for the comparison group. Okay, and the sample sizes aren't listed on this table, but I believe the authors stated the sample sizes in the text of the study. So you can go ahead and we'd go ahead and collect that information as well. And then once we have all the information, once we have the means, the standard deviations, and the sample sizes, we can go ahead and apply those formulas that I just illustrated. So first, we'd estimate. This is the formula for the pooled standard deviation, the average standard deviation. So you plug in the sample sizes and the standard deviations, and you get an average standard deviation of 16.6. And then formula 2 there is estimating the effect size that is not correct, that is not bias corrected. So the mean of the treatment group, 5.8 minus 16.2. And then you estimate Hedges G using that bias correction that we talked about. And then the last thing you do is estimate the standard error. Okay. So I know this is a whirlwind. I think Terri mentioned at the top of her talk that she was running through some slides that take, and in practice, sometimes up to a year to implement. In this case, calculating effect sizes for each study often takes just as long, and we could spend a lot more time on each family. But in the interest of time, I think we'll keep going with the whirlwind tour of effect sizes. And I'll pause for a second just to say, or we'll sort of switch lanes here and talk now about the R family. And the R family is, or the correlation coefficient family, is when we have studies that all measure continuous outcome measures, but now we're looking at the relationship, the quantitative relationship between two variables. And often this, the predictor variable here is also a continuous variable. So maybe you're interested in looking at the relationship between depression and anxiety, which is a pretty common, two pretty common measures in the literature. Or maybe you're interested in looking at the relationship between science achievement and math achievement. If you were interested in estimating or conducting a meta-analysis on those two relationships, then what you'd be collecting in terms of the effect size from each study is the correlation coefficient. I'll also mention that there, just like in the D family, there are lots of different variations on the correlation coefficient. For the most part, I think the literature is stuck pretty much to collecting and synthesizing bivariate correlations, but it's also possible to estimate different types of correlations and then synthesize those correlations. And so it's becoming more popular and common to estimate a partial correlation, which is from regression models or analyses that use the general linear model. And those partial correlations control for the relationship of other variables as well. So going back to our example of the relationship between math achievement and science achievement, maybe it would be possible to calculate a partial correlation if the authors controlled for other variables while looking at the relationship between those two variables of interest. So maybe you're interested in the relationship between math achievement and science achievement while controlling for the students' socioeconomic achievement, or maybe their motivation to learn or to be a part of, to learn math. If you're interested in doing that, then you calculate a partial correlation. So like in all things, the more you get into something, the larger and more expansive the world becomes, and it's no different for this correlation coefficient family. But if you are interested in looking at the bivariate correlation itself, the most common and the most simple effect size that we could estimate, you're in luck, because the information that you need to collect from the studies is quite minimal, and there's actually not a lot of calculations that you need to do as well. So if you're interested in doing a synthesis on the the correlation coefficients, then you're going to collect the correlation itself, which is, for the most part, usually presented by the authors, and then the correlation sample size. And then as we illustrate on the slides here, and that's it. Those are the two pieces of information you need, and they're pretty simple to collect. So we do a little bit of calculation on the back end. So after we've collected all of the correlations, in order to be able to synthesize the correlations in one model, we transform the correlation into what's called the Fisher's Z. And there's a little formula here to do so. And then once we transform the correlation into a Fisher's Z, we can then estimate the standard error of that Z. And again, the formula is pretty dang simple. In this case, it's just 1 over the square root of the total sample size minus 3. And that's it. Now you are all set up to conduct your meta-analysis and synthesize correlations. I told you it was a whirlwind. So we'll keep moving on with the different effect sizes here, and we'll come to the odds ratio family, and it's ish because just like the past effect sizes that can be calculated, there's lots of different ways to think about the estimation of the odds ratio and its variance. But the common factor among all the different types of effect sizes you'd estimate is that the outcome of interest here is a binary variable. So either you graduate or you don't, or you pass a test or you don't, or you get arrested or you don't. That's the key to this odds ratio family, is that all of the outcomes are binary or dichotomous, ones and zeros. And then the other characteristic is that your predictor variables or your independent variables are also among two groups. So commonly this is represented with the treatment and control group or the intervention and comparison group. And then we're going to look and we're going to count up how many participants from the two respective groups satisfied those two outcomes. So how many participants in the intervention group graduated from high school versus how many comparison group members graduated from high school? How many people in the treatment group passed or achieved some number on a standardized achievement test? And how many people in the comparison group achieved that value? Okay, so how do we actually do this? So odds ratio, as it sounds, is literally the ratio of the estimation of the two odds of success. So you're literally going to calculate the odds of treatment success. And in this case, we're looking at the success and failure rates of passing some test. And that's where this example comes from. So there's, in this particular example, there were 19 total students in the treatment group, 18 total students in the comparison group, and five of the 19 individuals in the treatment group passed the test, and 6 of the 18 passed the test in the comparison group. So we estimate the odds of success for each of those two groups, which you can see on the right-hand side here. And then we divide the odds of the treatment group divided by the odds of the comparison group, and that gives us our odds ratio. The odds ratio is a little bit challenging to interpret, but in most cases, a value that is above 1 means that the intervention was successful, means that the people in the treatment group were more successful, had a greater proportion of success. And if the odds ratio is less than 1, it means that the treatment group did worse compared to the comparison group. So in this case, we'd say that the treatment group did just slightly worse than the comparison group. So just like the last two effect sizes, we're going to calculate the odds ratio, and then we also need to calculate the odds ratio of standard error. To do that, again, we need to transform the odds ratio into a slightly different value in order to calculate the standard error. And so to do that, we're going to take the log of the odds ratio. And then once we do that, the standard error of the log odds ratio is a very simple calculation. And it's just that we represent each of the 4 cells of crossing the treatment group, the comparison group with the success and failure rate. We represent those as A, B, C, and D. And then we take 1 over each of those cells, add all that up, and square it, take the square root of that, and that's the standard error of the odds ratio. So for each effect size, it doesn't matter what the type, again, you're going to estimate that effect size, and you're going to follow it up right away with an estimate of the effect size, the standard error. Okay. The last thing I'll say before moving over to the R programming is that it's quite common that within your set of studies that are included in your meta-analysis, that a few of them have a dichotomous or binary outcome, and the rest have a continuous outcome. So a common question is it permissible to transform the odds ratio into a D or vice versa? And typically the simple answer is yes, you can do that. The underlying assumption is that in both cases, in the case of the binary outcome and the continuous outcome, that the outcome construct is the same. So as long as the binary outcome was a measure of achievement, and the continuous outcomes were all measures of achievement, it is likely permissible to transform the odds ratio into a D. Now, there's lots of different transformations available, and the transformations largely depend on the type of outcome data that the authors used and collected, and how that information was reported. And there is a really large body of knowledge and work that has been done that helps meta-analysts transform whatever data they have into the effect size that they are going to need for their meta-analysis. I've done. There was a gap in the literature back in 2016. Some of the most common transformations, like an R into an odds ratio and vice versa, hadn't been completely laid out at the time. And so I wrote a little open access paper with my colleague at 3IE, Berta Snizelbit, and you can go to this link or go to the Campbell library and find it. And there's a nice little table that shows all of the various transformation. So that's transformation. The good news, though, and this is a great thing to end on. The good news is, like I said at the top, from a very practical standpoint, you should rely on the work that has been done before you in programming languages to estimate all of your effect sizes. It's becoming exceedingly rare for anyone who's doing a meta-analysis to actually ever have the need to use any of the formulas that I just presented. And the reason is because all of it is readily available for you in R, in Stata, to some extent in SPSS, in one-off programs like comprehensive meta-analysis and RevMan and some of the other programs that are available out there. And what I mean by that is the data set that you create, after you collect all of the means, standard deviations, and sample sizes, once you have that for every single study, you tell the programs where that information is located in your data set, and the programs calculate all of the effects for you. And if you want, you can even use those programs to transform your effect sizes from one metric to another. And that that too is very simple. I don't have it on the screen right now, but it's a very simple procedure to do so. In the R language, there are little packages, what R calls packages, which are basically groups of functionality that allow you to do some analysis. And there are many packages that will do meta-analytic calculations, but by far the most popular package in meta-analysis is a package called metafor. And it's a great package, and I highly recommend looking it up, and I highly recommend taking the package creators classes. If you Google metafor, you'll find them. But I just wanted to show exactly what metafor, how you use metafor in practice. And so the lines starting with dat_BAI, that E, where it says ESCALC, that's effect size calculation, right? And that's the function that you use to estimate the effect size. And all we're saying here is that this measure, where it says measure, you're saying what type of effect size do you want to calculate? In this case, we're going to calculate the standardized mean difference, and you represent that with the SMD in the quotes. Moving on down a line, you tell R where the means of the two groups are located. So M1I equals is saying where's the mean of what's the column name of the mean of the first group. And in the case of this example, my data set, the column name was mean_TX, and then M2I is where's the mean of the comparison group. So there it is. The line down from that is SD1I, where's the standard deviations of the two groups. N1I is where the sample size is for the two groups. And then data equals means what data set should I be looking for? And then append equals true says, should the metafor package put two more columns on the end of your data set that represent the effect size and the standard error? When you say append equals true, it'll put those two columns at the end of the data set for you. And that's it. You run this script, and it doesn't matter if you have 2 effect sizes or 200, it will run it for every single one as long as you have that data, and you're done. Which is good, because I think, I don't really have a summary slide. So I'll just end by saying, if you take nothing else away from this lecture, it's that when you're doing a meta-analysis, you need to calculate an effect size for each study. An effect size is a measure of the magnitude of the relationship between the two variables. Often that relationship is either the magnitude of the treatment effect, so how much did the treatment and the intervention group differ, or the relationship between two quantitative variables, two continuous variables like math achievement and science achievement. And so you're calculating an effect size from each study, and you're going to calculate a standard error from each study. And once you do that, then you can compare the effects across studies, or you can combine them in a meta-analytic model, which Ryan will show you in a minute. And that's it. It's as simple as that. It takes a really long time, but it's a fun process. And I think I'll stop there.
All right. Thank you so much, Josh. Excellent presentation. And you're getting all kinds of accolades between the chat and the questions. Just one question before our break. Can you say a little bit more about when you have a small sample in studies, and then you're calculating the effect sizes?
Yeah. So that goes back to the estimation of Hedges G, which is the small sample correction that is applied to the standardized mean difference. And essentially what Larry Hedges' paper said was that in really small sample sizes, so sample sizes that are less than 50, the effect size that you'd estimate without the bias correction is just a little bit too big. It's just overestimating that relationship. And And so what the hedges G effect size attempts to do is correct for that small amount of what we refer to as bias. So it's slightly too large, and that correction decreases that standardized mean difference just enough so that the bias is eliminated in small samples. Now, for samples that are larger than 50 or certainly 100 to 200 participants, that correction is very, very, very small, and it makes very little difference to the standardized mean difference. It's only impactful when the sample sizes are pretty small.
Great. Thank you. All right, well, we're going to take a break for about 5 minutes here to allow everyone to stretch and pull away and all, and come back at exactly 20 before the hour, or so it's 2:40 Eastern time. I just want to say to everyone too that this recording will be available on the AERA website and also the Peers Data Hub site, but give us a week or so to get that together, but it will be posted. And I put into the chat and things too, and from that the email message you received earlier, how to access the slides. So let's take our break.
Okay, I think we're going to get restarted here, or at least I'm going to launch us back in, so make sure we can finish on time. So what I'm going to cover here is the last piece of the presentation. So Josh and Terri kind of walked us through the first two big phases of meta-analysis. And so Terri covered kind of how we're going to identify studies for the review, and Josh showed us how we're going to extract information from those studies that we need for our synthesis. And I'm going to walk through some of the basics of how we would combine those effect sizes that Josh was just talking about. And so there are two big kind of families that I'm going to focus on, again, at kind of a high level. And if you come to one of our additional trainings, we'll take a deeper dive into these. The first is the fixed effects model, and the other is the random effects model. And we'll kind of navigate together kind of what it means to use each of these approaches, and what some of the implications might be for a meta-analysis. And so I'm hoping, just like with Josh's presentation, you all take away that this is a fairly accessible set of mechanical processes that are used to combine all of these effect sizes. The basic part of it is a weighted average, but these two different types of modeling strategies are going to have implications for how we characterize the weights, and what kind of information goes into those weights. So we'll get into that. All right, so first step, this is something that we get pretty frequently in our meta-analysis courses. How do we choose between a model? Either a fixed effects or a random effects model. And the basic advice that I think we would all give to our students and course participants is that the choice between a fixed and random effects model needs to largely be a conceptual choice. So something that's tied to a theoretical understanding of how those effect sizes might be distributed in the population of the research that you're studying. So if you're looking at a particular, say, science curriculum, you may have a content expert on your meta-analysis team who can give you some insights into how implementation of that curriculum might vary across contexts, or for different types of learners, or may be more or less effective for certain types of outcomes. Another consideration is, or maybe a simple question to kind of ask yourself as you're getting started is, when you see variation in effects, is it plausible that the variation is simply due to having different samples of participants, or is it something else? If all we think if the only source of variation is sampling variation, then maybe a fixed effects model might be appropriate. If we think there are other potential sources of variation that we can anticipate, or maybe we may not anticipate or fully understand, then we're probably more likely to be in a random effects framework. Another consideration with the choice of the modeling strategy is the amount of data in hand. So we're going to kind of work through some simple examples in this presentation, again, at a high level. Small data examples, but when you have only a few studies in hand, say fewer than 5, there may be some difficulty in estimating a key parameter needed for the random effects model, and I'll circle back to that a little bit later on. But that also reminds me of a key distinction that we can make between the fixed effects as a plural modeling strategy and the random effects modeling strategy. So we'll come back to that in a moment. So let's start with the fixed effects model. So in the fixed effects model, we're again really kind of thinking that there is one source of variation in the effect sizes. And that's just sampling variation, right? So we see different effect sizes because there are different samples of participants who are included in those different studies that are generating your effect sizes that Josh just talked about. And so if that's the case, we would expect fairly minimal variation in the average effects across these studies. So this little diagram here is something called a forest plot. So we can see that not all of these bars, these horizontal bars that might represent confidence intervals, they're not all the same size. Some are smaller than others, so those are probably larger studies. But in general, they're kind of telling us the same story. The formula for calculating the meta-analytic average under a fixed effects model is pretty straightforward. And that's going to involve the meta-analytic weights. And in meta-analysis, we use inverse variance weights. And for the fixed effects model, again, the only source of variation is that sampling variance. So those standard errors that Josh just showed us how to estimate, those go into the denominator of our weight formula here. So 1 over our squared standard errors, that's going to be the weight. We multiply those by our effect sizes and divide by, and sum those up and divide by the sum of all of the weights across the effect sizes in our data set. And that gives us a weighted meta-analytic average under the fixed effects model. So that's pretty straightforward. And I should note here that this is the basic framework for getting a meta-analytic average. We're going to tinker a little bit with what goes into our weights and how we conceptualize that, but this basic weighted average formulation is not going to change. So this model is, like I said, it's helpful for synthesizing effects that are homogeneous. So they're all kind of giving us the same signal about what that true population average effect size is. We only see differences or variation in effect sizes because of different samples. This model also suggests that the effect sizes are coming from pretty carefully scripted replications, maybe even within the same study. So you can imagine some kind of carefully designed laboratory type replications that allow pretty minimal variation in the types of participants, the types of outcomes, the types of interventions or the variations in the interventions and the settings that are generating the data for this type of meta-analysis. So if those kind of best describe the studies going into your meta-analysis, this might be a useful starting point. And like I said before, if you've only got a handful of studies, say fewer than 5, practically you may be constrained to using this type of a modeling approach. And again, I'll come back to this once we get into random effects. So the random effects model, again, has the same general formulation here. So it's the sum of the weighted effect sizes divided by the sum of the weights. But our little description here of what the effect sizes might look like in a random effects model is different. So we not only see variation in kind of the precision of each of the effect sizes, the width of those confidence intervals, but the midpoints of those confidence intervals where the effect sizes would live, those are also varying, okay? So in the random effects framework, our inverse variance weights have 2 components. Our sampling variance that Josh introduced to us, which is largely a function of sample size, but also our between study heterogeneity that we can estimate a few different ways. And this is just a constant. So each of the effect sizes has its own sampling variance, but then there's a constant estimate of the between study variance, okay? But the basic estimation procedure, that weighted average, remains the same. So because we're going to apply 2 different sources of variation under the random effects model, the standard error for the mean effects when that between study variance is not 0 is going to be larger than the standard error for an equally sized fixed effects model, okay? Right, because we don't just have sampling variance, but we have between study variance. And again, unless we have these pretty carefully scripted laboratory type conditions going into generating effect sizes for our meta-analysis, it's hard to conceptually rule out a random effects model. And Terri kind of described typically sized meta-analyses in education research being anywhere from 20 to 30 or 40 studies. It's usually implausible that those are homogeneous distributions of effect sizes, meaning a random effects model is probably the more appropriate model to start with. So the choice between the fixed effects model and the random effects model, like I just indicated, has implications for the precision of the meta-analytic parameter estimates that you may be interested in, like the study average. Again, the general formulation is that the weights are a function of the effect size variances, but how we conceptualize those variances is going to differ between the fixed and the random effects models. So holding everything equal, larger sample sizes are going to equal smaller variances, larger weights, more precision in your meta-analytic averages. We can do a bit of a side-by-side comparison of these two weighting strategies here and see what happens as our between study heterogeneity parameter tau squared increases or decreases. So again, under the fixed effects modeling strategy, we only have our single variance component and our denominator for our weights. But let's say we have a tau squared estimate here that's pretty small or even near zero. In that case, the random effects model converges on the fixed effects model, right? And each study is going to be contributing to that meta-analysis roughly proportional to their sample sizes. In the case that tau squared is large relative to the sampling variances, that's actually going to have an equalizing effect on the weights for each of the studies. So studies that were larger are going to kind of have a bit of a downgrade in their contribution to the meta-analysis, and smaller studies are actually going to get upweighted a little bit, right? Because we're adding that constant. And so it kind of pulls all of these effects into the center a bit as a bit of an equalizer. So again, we're going to walk through an example here of generating a weighted mean effect size. The basic formulation is just a weighted average. The only distinction here for getting a mean is how we define our W terms, our weights here. But I'll walk us through here with a little fake data set. These are all kind of standardized mean difference family effect sizes, or the D family effect sizes that Josh was talking about earlier. But the same set of procedures that I'm going to walk us through here would hold if we had log odds ratios or correlations or something else. The variances would obviously change. So here's a fake data set with 4 studies. I've just called A, B, C, and D. We have their effect sizes here. That's ES sub I. We have their sample sizes. That's just N sub T for the treatment group, N sub C for the control group. The variances using our variance formula that Josh introduced to us. And then the weights, which are just 1 over the variances. So these are fixed effects weights that we'll start with. And that's all of the information we need to get going. So, whoops. So now we can count with that, with our variances and our effect sizes, we can calculate our average. So we sum up the weighted effect sizes, so the sum of the weights times the effect sizes, and divide that by the sum of the total weights. And that ends up giving us an average effect size under a fixed effects model of negative 0.13. But the nice thing about meta-analysis, instead of just taking a simple average or even just a weighted average of our effect sizes, is we can describe our uncertainty or the precision around that estimate. So we can now get the variance using the weights that we just applied to get our average. So the variance for the average is just going to be 1 over the sum of the weights in this case. And so that's just 1 over about 203.3, which is about 0.005, which means we have a standard error of about 0.07. So we have a mean of about negative 0.13 and a standard error of about 0.07 for that estimate. So calculating the mean and the variance around the mean, that's an important step. But another important step, regardless of the modeling strategy that you decide is appropriate. So even if you're pretty darn certain that a fixed effects model is appropriate for your data, it's a useful exercise to scrutinize that decision a little bit. So even if you don't think there's a whole lot of heterogeneity in your data, it's useful to interrogate it. And so we're gonna walk through how we conceptualize that heterogeneity term and test it, and at least test a basic hypothesis about whether or not those effect sizes are truly homogeneous, or whether or not they truly fit that fixed effects model that we just used. One other thing that I skipped over here is, in addition to describing the standard error for the mean effect, we can also use that standard error just like we would in a primary study to get a confidence interval. And so this is a simple formula using a standard normal distribution with an alpha level, say 0.05. We can use that information to back out a 95% confidence interval. So just using our effect size, subtracting off 1.96, which is the critical value on a z distribution, and multiplying that by 0.07. And we do the same thing for the upper bound version, except we add 1.96 times 0.07 to our average effect size. The other thing that we can do, so that confidence interval already tells us something about whether or not the average effect is different from 0, but we can formalize a z-test. So we take the absolute value of our effect size and divide by the standard error that we estimated, and we get 1.78. So again, comparing that to a critical value or 95% critical value would mean that we would retain the null hypothesis in this case, that the average effect is equal to 0. Again, this is just a fake little example. Okay, so now let's talk about effect heterogeneity a little bit. Effect heterogeneity, I think, is one of the most interesting and useful aspects of meta-analysis. So meta-analysis gives us an opportunity that we don't typically have in primary studies to kind of see how effects, intervention effects especially, vary across populations, across outcomes, across settings, etc. So we're going to talk about testing for heterogeneity using something called a Q-test, and then also describing the magnitude of heterogeneity using that tau squared term that we saw earlier. So statistical tests address whether or not the set of effect sizes you have in hand are at least homogeneous enough or whether or not the homogeneity or heterogeneity we see in the set of observed effect sizes is due to chance, basically. And that's what the Q-test is basically going to test. Here's the formula for a Q-test. It's really got these two terms. The first is, and none of this requires new information that we haven't seen already. We have our weights, so we're going to sum up the weights times our squared effect sizes. And then we're also going to sum up the weights times our effect sizes and square that and divide it by the sum of the weights. And that's going to be our Q. Larger values of Q are going to indicate that there's more heterogeneity in the observed effect sizes. Smaller values will be indicate more of a homogeneous distribution of effect sizes. Then we have our tau squared estimate, which can be derived from Q in this presentation. So if we have Q in our degrees of freedom, which are simply the number of studies or number of effect sizes in this case, -1, and we already have our weights and our squared weights, and we can get our estimate of tau squared, which is the variance of the population effects. And I'll walk through an example of doing that too. All right, so let's revisit our toy data set here. And we have added a column here that's our squared effect sizes times the weights, our fixed effects weights in this case. If we sum those up, and we have the sum of our weights and the sum of our weighted effect sizes, we have all of the pieces we need to calculate our Q statistic. So we plug those pieces of information back into our formula for Q, 9.93, -25.33 squared, and divide that by 203.33, and that gives us a Q of about 6.78. We'll come back to whether or not that's statistically significant or not. But first, I wanna just quickly address tau squared. We only have 4 studies here, so this isn't a great candidate for estimating tau squared or estimating tau squared with a lot of certainty. We're going to tend to overestimate heterogeneity in smaller and meta-analyses with fewer studies and smaller sample sizes. But this would give you a sense of how you would do it anyways. So you plug in our Q, our degrees of freedom term, and the other weighted effect sizes terms that we saw earlier, and we get a tau squared of about 0.03. The square root of that is the standard deviation, or the estimated standard deviation of the population effects. And that's really interesting information to focus on for a meta-analysis. In fact, that's where I think most of the action is in meta-analyses. It's not just in the means, but it's in the variation around the means and what explains that variation. So again, this is likely an overestimate, but if we pretended for a second that it wasn't, we'd be really interested in what was driving that 0.03 or 0.17 in there, and we'd be really attentive in building our code books to identify study features that are related to that. Okay, so again, Q just tells us it's an index of the degree to which variation is different from chance. Tau squared is the magnitude of variation. So you can think of tau squared really as kind of the effect size version of Q in some ways. In fact, I think it's always a useful descriptive piece of information to include in meta-analyses, especially using random effects, obviously using random effects. Okay, so back to Q for a minute. So we can test whether or not Q is statistically significant, that is, whether or not the effect sizes are homogeneous or not, using a chi-square distribution with the number of effect sizes -1 degrees of freedom. So if we plug that information, we can identify a critical value of about 7.82. Our Q statistic was smaller than that. So in this circumstance, we would not reject the null hypothesis that those effect sizes are homogeneous. This is worth cautioning though. The Q test, especially in examples like I have here with few studies, can have pretty low statistical power, which is really the main reason you don't want to use it to determine whether or not your study effect sizes are homogeneous or heterogeneous. That should be a conceptual decision made earlier on. Using a random effects framework, even in this case, will result in a more conservative estimate of the precision of the average effect. So if we were to run a random effects model, which is to say, if we were to take this 0.03 and we were to add it back into the denominator of our weights, we'd end up seeing an average effect size that's about the same, -0.13, but our standard error would blow up to about 0.11. So again, we've probably overestimated tau here. Oh. Oh dear.
While we're waiting for Ryan, I just want to remind everyone that this recording will be posted on the AERA website. Please just give us a week or so, and that we'll have this and have this readily available. You know, a lot of that we are asking those questions in the chat.
Do you want to ask a question, George? We can, while we're waiting for Ryan to come back.
Well, yeah, there was maybe one question that you can get back to. I'm going to go back to some things in your presentation, Terry. Can you speak to a little bit about using artificial intelligence or AI as you're conducting your literature search?
Yeah. So, so there have been some great. Let me put my video back on. There have been some great innovations using machine learning. Right now, I think the most developed version of this is for screening. So I mentioned Abstractor and Rayon, and these are two programs that can really help you screen titles and abstracts. So you upload your titles and abstracts into these programs, and then as you start screening studies in and out, the program is running a machine learning algorithm in the background, and it's figuring out how to what studies are going to be screened in. And then it keeps rearranging your studies so that the most likely ones float up to the top. And in addition, additional functionality of these programs is also to help you organize the process. There are other developments in the works for coding, but you can imagine that this is a really difficult thing to do with machine learning, to actually code studies, using natural language processing. And there have been some developments in thinking through how to help get the right kinds of search terms for particular kinds of systematic reviews. But those are all, again, sort of in the works and not quite ready. So I see Ryan's back, so I will stop now.
Terri, can you hear me?
Yes, I can.
Okay, great. Sorry about that. I had a brief technical error with my audio, went out. Thanks for jumping in. My video is also off for the minute, but I'll see if I can get that back on. Okay, so so we were just talking through quantifying heterogeneity using Q and tau squared, and how we would test whether or not tau squared, here we go, sorry, whether or not Q was statistically significant or not. And so in our little example, we had a Q of about 6.78 or so. The critical value is 7.82. We don't reject the null hypothesis. That's not sufficient information for us to fully rule out a heterogeneous distribution. And like I said, if we were to incorporate that tau squared estimate that we had earlier, which is about 0.03, and add that into the denominator of our weights, our meta-analytic average standard error goes up to about 0.11. So if nothing else, if you find yourself constrained to a fixed effects modeling approach, or you think it's well justified, even with a lot of studies, it's, I think, always a good idea to evaluate the sensitivity of your key parameters, like the means, or if you get into meta regression later on, how sensitive that assumption is to possible heterogeneity. Okay. All right, so just like Josh had in his, I've provided a little bit of R code here, which is, again, tapping the metafor package as well. So metafor is really becoming largely a one-stop shop for all things meta-analysis, effect size, computations, certainly synthesis, data visualization, et cetera. And it's really fairly straightforward. I've broken it out onto multiple lines of code here, but really it's kind of one line of code. So if we were to load our effect sizes into an object called ES, these are the same data that we had up before, and create another variable called V, which are the effect size variances, we have everything we need to run a meta-analysis, either fixed effects or random effects. So we use the function called RMA, and we identify our effect sizes. So YI, it's going to look for argument YI, and we're saying those are equal to ES, our effect sizes here. VI, it's looking for the variances right here, and ignore this M2I, that's a holdover from some of Josh's code, I think. So we'll edit that out. And then specifying the method. So it's really, you need three pieces of key information: your effect sizes, the variances, and the method, which is going to be whether or not you run fundamentally, it's going to be whether or not you want to run a fixed effects model or random effects model. Within the suite of random effects models that you might run, there are different estimation procedures to get at tau squared. But we covered some of that in our other trainings. So if you were to run all of this, you should get the results that are pretty darn similar to what we did kind of manually up above. And then for a random effects model, everything is the same here. The only thing I've changed here is the method, which is DL in this case, which is the Dersimonian and Laird, or method of moments estimation procedure for tau squared. And that's just kind of that tau squared that's derived from Q that we could largely do by hand. There are other estimation procedures that are useful, especially with large numbers of studies, like restricted maximum likelihood, that are also really useful. Okay, so this is kind of the cliffhanger at the end of the presentation. If you come to one of our other trainings, we really dive into this idea of what to do about heterogeneity. In fact, our training institutes are largely kind of focused on doing meta-analysis for understanding heterogeneity and effects. And we go through kind of a number of recommendations for how you would handle it. Just like with regular studies and studying associations and variation, this is a model building activity. And so we want to try to build conceptual models that are grounded in those UTOS or PICOS frameworks that Terri was talking about, that provide substantively useful results to consumers at the end of the day, and also tell us something about why these effect sizes are varying the way they do. So just like we would build multiple regression models for our primary studies, where we try to simultaneously adjust for multiple things, we apply the same sort of logic here in meta-analysis for all the same reasons that we would in a primary study. So we aren't going to get into meta-regression today, but I think it's one of the most important and useful aspects of meta-analysis, which isn't to downplay the importance of aggregating studies to estimate a mean effect. That's important, but almost always, and certainly in education research, we see non-trivial heterogeneity in effects that warrants investigation. Our trainings largely focus on R and implementing meta-analytic models in R, but certainly depending on the scope of the meta-analysis, the size, you can use other software that are readily available and typically used: Excel, SPSS, Stata. We definitely don't recommend doing it by paper and pencil. So if you're thinking about doing that, just knock that off your list and try out one of these other options. There are also some programs out there that are specifically designed for meta-analysis, like comprehensive meta-analysis, or CMA. That's really kind of the entire enterprise is for synthesis. And of course, you have the metafor package within R, which is highly focused on this. And here's some other resources to learn about systematic review and meta-analysis. But I think, am I going to pass it back over to you now, Terri, or do we want to do questions and then close out?
I can just quickly do that if you just, I'll just tell you when to turn the slides. So I just wanted to let you be aware that we have a almost, I think it's 3 to 4 hour workshop that is in the AERA 2020 Virtual Research Learning series. So you have the link there. We are also doing an advanced meta-analysis course virtually for the 2021 annual meeting. The sign up is there. We have some resources right now focused mostly on R and using and doing meta-analysis in R on our IES funded Meta-Analysis Training Institute website, which is there at the bottom. And then Brian, can you just go one more slide? I just have one more slide here. And then I also wanted to tell you that we are actively working on a more elaborated website that'll have a lot of resources for looking at the entire systematic review meta-analysis sort of pipeline. And we're going to be posting that soon. It's going to be called the Meta-Analysis Learning Information Center. And if you just check back on our NSF-funded Modern Meta-Analysis Research Institute website, the link is there. In the next couple, I would say realistically in the next month or so, you'll be able to get some resources there. They're mostly, there's some quick video tutorials right now up that will help you do ESCalc and so forth. And then also feel free to email us at our Modern Meta-Analysis Research Institute website there at mari@gsu.edu. So I will stop. And we have a few minutes for questions, so go ahead, George.
Okay, great. Thank you. Thank you. We had a couple of questions that pertain more so to Ryan's presentation. Ryan, can you speak a bit more about moderator variables? There was someone asking, well, when there are moderator variables, how do you account for those?
Yeah, so yeah, unfortunately, we couldn't get into moderator variables today, but like I said, I think they're one of the most important and interesting aspects of meta-analysis. Presumably, when we're starting a project like this, we have some substantive insight into what might drive variation effect, like implementation fidelity, like types of learners who may benefit more or less from certain programs, and types of outcomes that may be more or less responsive to those programs. When we have those ideas, we build them into our codebook. And we build them in and we plan analyses around those as a way to study how they may systematically tell us about variation in effect sizes. So one of the things that I like to do for a lot of my meta-analyses is that tau squared parameter that we had. You can kind of treat that as like, turn that into a variance explained sort of parameter, like how much of that variable or how much of that parameter estimate can I explain with my moderator variables about the participants, about the interventions, about the outcomes and the settings, or the comparison groups. And that's, I think that's a critical part, especially when you're doing random effects models. Keep in mind, when you're doing random effects models, you're already kind of acknowledging that there are multiple true values. And so getting at what's the conditional effect for certain types of learners, for certain types of outcomes and certain types of settings becomes all that more important.
Great, great. Thank you. That was probably our most of the questions were around some things around variables. I think you covered most of the things. I know in interest of time here, And I know we also have some of the, not the other trainings that are going to happen at AERA at the annual meeting virtually, and as well as things we've done in the past. There have been some people have been asking general questions about doing doing qualitative meta-analysis. Can one of you, one of the instructors, speak to that, to qualitative?
Yeah, I think I can do that, Ryan. So, this is a whole area of, of ongoing research right now. Quickly, if you want to send me an email, I have at least a page of resources I can send out to you. But I think at some point, maybe Felice, there's going to be a qualitative one, because there is an NSF funded grant there.
Right. It's one. Terry, I was just going to say it's one in the series of 11. I'm not exactly sure which month, but if you could share your resources, we'll also post the other PIs.
Okay, that sounds great. I will do that. And if you, again, if you email us at mari@gsu.edu, I can send out that the one page that at least can get you started. So.
Great, thank you. And I know so many people have posted questions that sort of referencing some of their own work and their own studies. I'll emphasize that we will come back together in a meeting mode here to discuss where you can ask those kinds of questions. So I'm going to share in my screen here. I think this will help you to see where you can our link to this here.
So George, just to be clear, we're going to get out of this meeting and we're going to go into a different one, right?
Into a different one, right. So
okay.
I'm hoping that everyone can you?
George, you want to sort of, for those who would be unaware that we're doing this extra special half hour, do you want to sort of?
Yes. So as I was saying to everyone, what we're going to do is we're going to come back in a more of an informal discussion in a meeting mode where we'll all see you and you can see the instructors, and you can ask those burning questions in a very informal discussion with the instructors in the group discussion. So we know that because there were 1,500 or so people on today, so this will make this a lot less formal. You can ask those questions you want. We'll be on for about a half hour. So
this is the of course we're drug-free workplace. So with that footnote, I'll say this is like you might say after a course, let's hang out at the bar, okay? But we'll say let's hang out at the milk bar. So any of you who are on and want to kind of join and chat informally, just follow that other Zoom, and it'll be an opportunity to just have a conversation together if you have the time and want to come.
Yeah, and I'll post this in the chat as well, so that people can see that. Yeah, I want to thank our instructors today, though. This is an excellent, just an excellent presentation. Okay, so just sort of share this here. Just do I still have chat?